Hello, my name is Miriam,
And my name is Matthew.
and today we'll be presenting our article, which investigates the cryo-EM structure of the entire mammalian F-type ATP synthase. So just to introduce the enzyme, ATP synthase is comprised of membrane-bound protein complexes responsible for ATP synthesis, the utilization of proton motive force, and for establishing the proton motive force from the energy released by ATP hydrolysis. The hydrophilic F1 domain is present in the mitochondrial matrix and is responsible for the ATP synthesis. The membrane-embedded F0 domain involved in proton translocation consists of a rotating C ring. These two domains are connected by a rotating central stalk inside of the F1 domain and a stationary peripheral stalk. Additionally, the enzyme is also involved in the formation of the permeability transition pore, which is responsible for triggering cell death. The F0 is comprised of C subunits. These are arranged in a ring to facilitate proton translocation as it rotates and protons enter the enzyme from the intramembrane space of the mitochondria. This experiment focuses on solving the structure of mammalian F1, F0 structure, as there was limited knowledge on its F0 domain. The study also suggests that the C ring is the location of the permeability transition pore, which triggers cell death when opened by the accumulation of calcium ions or intense oxidative stress. The role of ATP synthase in the pore's opening and the discovery of the lipid plug in the C ring are further explored in this article. The structure and function for the V-type ATPase and F-type ATPase were known when the study was published, but there was no knowledge on the mammalian mitochondrial ATP synthase. Through crystallography, the F1 subcomplex structure had previously been revealed, but due to limited resolution, the subunits were either modeled incorrectly or misplaced. This left most of the structure of the membrane domain unknown, and the aim of the researchers was to solve the structure of the mammalian F1 F0 ATP synthase. Cryo-electron magnetic microscopy is a technique used to develop detailed and high-resolution images of complex protein structures. In this article, atomic models of the membrane F0 domain and the mammalian F1 F0 subunit were made possible through this technique. Cryo-EM begins with a sample of a protein and pure water. Liquid ethane is then used to freeze the samples so that the proteins are embedded in their natural state in a thin sheet of ice. An electron gun then fires electrons at the speed of light through the sample, which are detected by a camera. The camera then produces images of the protein molecules in various orientations. Next, computer places protein molecules in the same orientation into groups. Finally, a software program produces a high-resolution 3D composite image of the protein in which individual amino acids can be visualized. The rotation of the F1 hexamer is driven by conformational changes undergone by the beta subunits as the ATP, ADP, magnesium ion binding sites are filled during ATP hydrolysis. Each full rotation will take each beta subunit through the conformational states of beta TP, beta DP, and beta E, with the force driving the rotation coming from the beta subunits as they move through their conformational states.
To elaborate a little bit more on these specific findings of this article, I'll talk first about the first part of the lipid plug removal process. It involves calcium binding to the beta subunits instead of regular magnesium ions. Calcium ions have larger ionic radii than magnesium, which is traditionally associated with F1 binding and binds to the beta subunits in order to bring out greater conformational change in those beta subunits. The increased conformational changes resulted greater than normal pushing and pulling forces on the B subunit of the peripheral stalk. The peripheral stalk is connected to the F1 crown via oligomycin sensitivity conferryl protein, or OSCP, and needs to be flexible enough to withstand the wobbling forces generated by the F1 ATPase. The next part of the lipid plug removal involves the binding of cyclophilin D, or CyPD, to OSCP. CyPD has the effect of stiffening the OSCP PS assembly, making the complex holding the lipid plug more susceptible to forces. The larger than normal F1 conformational changes from the calcium ATPase coupled with the increased susceptibility to generated forces from CyPD stiffening of the OSCP PS assembly result in subunit E being pulled away from the C ring. This also results in the lipid plugs being pulled out. As the lipid plug on the intermembrane side is pulled, water molecules flow into the ring and work to destabilize the lipid plug on the matrix side. This first stage of the process is reversible. Finally, as the hydrophobic interior of the C-ring is destabilized, the C-ring is widened by about 2 to 3 nanometers and is followed by F1s detaching completely. This results in the complete opening of the conductance pore, and this final portion is irreversible. So the major finding of this experiment was that subunit E is connected to a lipid plug within the C-ring. This is important because with one anchor at F1 via OSCP subunit and another anchor being at this new C-ring via subunit E, the ATP ACE is able to rotate in a wheel-bearing system. This C-ring anchor further increases the stability of the ATP ACE and another role in the protein translocation pathway. In ATP synthase, the protein translocation pathway describes when the C-ring makes a 180-degree counterclockwise rotation, starting from the IMS, and the synthesized protein is released into the matrix. Without subunit E, this rotation might not happen so smoothly. In this image, you can see the protein carrier, E58C, in the middle of the C-ring. When E58C is protonated from the IMS, E58C rotates within the ring, then releasing the carried protein into the matrix. Another finding in this study was that the addition of magnesium monomers in the mitochondria did not affect the F1, F0, and PTP opening physiologically. However, when calcium monomers were added to the subunit E and C ring, they had actually distorted in multiple steps until the C ring was no longer visible, thus allowing for protein movement. One final important finding from this experiment was that although both the dimer and tetramer formations of the F1, F0 domains were found, the primary structure was found in tetramer form. This finding serves to indicate how the mammalian mitochondria membrane might truly be arranged in tetramer form in a typical physiological setting. These newly identified structures even serve to negate previous studies, as it had shown that many F0 subunits were not complete in their design. If you take a look at the image we have on this slide, the top row of images actually serve to display the tetramer form that was found as a result of this study. In conclusion of all this article, some major results include the conservation of the tetramer model in the presence of magnesium ions, which serve to indicate that this arrangement is reflective of a typical physiological arrangement in mammals. Next, the ATP synthase stator, or stationary portion of the protein, is anchored at 2 points, one being the known fixed connection to F1 domain via the OSCP subunit, and 2, the newly found connection to subunit E via the lipid plug, with both of these serving to allow rotation. Both of these results help to lead to the conclusion that subunit E is connected to a lipid plug in the C ring. There's a highly conserved lysine in mammals on the C terminal, which is proposed to interact with the head group of the lipid forming the plug. The binding of CyPD will stiffen the assembly, while CSA blocks the groove in order to prevent binding. This, in addition to conformational changes associated with calcium attachment, leads to a removal of the lipid plug. Some further research that could be investigated into using the results of this study may be made into the PTP opening and regulation, F0 dimerization and cristae formation, and finally, this development of drugs specifically targeting mammalian ATP synthase for uses in diseases such as tuberculosis, cancer, ischemia, and aging and neurodegenerative disorders. If you take a look at the image on the left, this is a model of a mammalian ATP synthase, specifically with inhibitory sites circled in red. As you can see, they're pointing to specific inhibitors that may be used to target those sites. And of course, these inhibitors serve a variety of functions, many of which may be used as treatments for the diseases mentioned on the right. Here is just a quick glance at most of our references used in this presentation. And now we'd be happy to answer any questions that you might have. Thank you so much.